Welcome to the Deep Dive. Today we're jumping into a really fascinating area, the mix of artificial intelligence and chemistry.
Yeah, it's a hot topic. We're using Mohammed Medakaber's document, Artificial Intelligence in Chemistry, sort of as our guide.
Exactly. And the mission for you listening, get a solid, quick grasp on how AI is shaking up chemistry. We'll hit some surprising applications, key insights, but, without drowning you in technical jargon.
That's the plan. And what's really driving this whole thing is just the sheer amount of data we have now in chemistry. It's exploded.
Right. Things like high-throughput screening, all the spectroscopy data, genomics, proteomics.
Yeah.
It's a data deluge.
It really is. And it's fantastic data, full of potential, but actually getting the value out of it. That needs new tools.
And that's AI, right? It comes in as this super powerful way to, make sense of it all.
The document really emphasizes this. AI is great at spotting patterns, hidden connections and all that data.
And predicting outcomes, optimizing processes, even automating some lab tasks that take forever.
Precisely. It can predict stuff, fine-tune reactions, handle routine tasks. So in this deep dive, we'll cover a bit of the history,
Okay.
some of the core AI techniques involved, why managing all this chemical data is such a big deal.
Yeah, the data side seems huge.
Oh, it is. And then we'll look at the cool applications, drug discovery, new materials, that sort of thing. And of course, the ethical side too.
Right. Sounds like a plan.
Yeah.
So let's rewind a bit. Where did this connection between AI, or at least computation and chemistry, actually begin? What's the history?
Well, the document traces it back to the early days of computational chemistry. Think about foundational methods like the variational principle, perturbation theory, the Hartree method, Hartree-Fock. These were like the first real attempts to calculate chemical behavior from first principles.
But incredibly difficult to do back then, I imagine.
Oh, unbelievably laborious. Before computers were common, these calculations were just monumental tasks. Really painstaking.
I can only imagine. So computers arriving post-World War II must have been transformative.
A total game changer, yeah. Suddenly, scientists could tackle calculations that were just impossible before.
Like what? Any specific examples?
The document points to the first ab initio calculations, that means from scratch, basically on simple diatomic molecules around 1951. That was huge.
Right.
And then came semi-empirical methods, like the Huckel method. These used some experimental data to simplify things, making calculations faster, trading a bit of accuracy for speed.
So finding that balance between what's possible computationally and getting useful answers.
Exactly. And all that early work really laid the groundwork for everything that followed. It set the stage.
Okay, so that's the computational chemistry side. Then machine learning itself starts evolving and finding its way into chemistry problems. What did that timeline look like?
Right, so the document maps this out too. It starts back in the 50s with the perceptron algorithm. Think of it as a very early, simple type of neural network for basic pattern recognition.
Okay, the very beginning.
Yeah. Then the 60s and 70s saw things like k-nearest neighbors, or KNN, and decision trees. These offer different ways to classify data or make predictions.
More tools in the toolbox.
Exactly. But then a really big moment came in 1986, the backpropagation algorithm.
Ah, I've heard of that. That was important for neural networks, wasn't it?
Hugely important. It basically gave neural networks an efficient way to learn from their mistakes, to adjust their internal connections. This suddenly made it feasible to train much deeper, more complex networks. It really revitalized the whole field.
Okay, so backpropagation kicks things into higher gear. What came next in the 90s and 2000s?
The 90s brought support vector machines, SVMs. These are really powerful for classification and regression, especially good with high-dimensional data. They use clever maths, kernel methods to handle complex patterns.
Right.
Then the 2000s were big for ensemble methods, things like random forests and gradient boosting.
Ensemble, meaning combining models.
Yeah, exactly. Instead of relying on one model, you build lots of them, often simpler ones like decision trees, and combine their predictions. It usually gives you much better accuracy and robustness.
Makes sense. Power in numbers.
Sort of, yeah. And then the 2010s, well, that was the deep learning explosion.
Right, that's when things really took off across the board. Images, language.
Exactly. Deep neural networks, networks with many, many layers, started achieving incredible results. And naturally, chemists started thinking, hey, how can we apply this to our problems?
So alongside these general ML developments, were there specific pioneering AI projects focused just on chemistry?
Oh, yes, absolutely. The document mentions a few key ones. Dendril, way back in 1965.
65?
Yeah, an early expert system. It used heuristic rules of thumb, basically to help figure out the structure of organic molecules from mass spectrometry data. A classic example of AI for interpreting complex scientific data.
Interpreting that spectral fingerprint. Okay.
Then there's ELISA, starting in the late 60s. This one was fascinating. It tackled retrosynthetic analysis.
Retrosynthesis. Planning a synthesis backwards.
Exactly. You give it a target molecule, and ELISA would suggest possible synthetic pathways step by step based on a database of known chemical reactions, like an AI assistant for planning how to make something.
That's pretty sophisticated for the time.
It really was. And then from the 70s onwards, QSR models became really important.
QSR. Quantitative structure-activity relationship.
That's the one. Using statistics and later machine learning to predict how active a molecule might be biologically, based purely on its structure. Hugely influential in drug discovery.
I bet. Finding potential drug candidates faster.
Definitely. Then fast forward to the 2000s, you see robotic synthesizers emerging, automated systems actually doing the experiments, mixing chemicals, optimizing conditions.
Taking the automation to the physical lab bench.
And more recently, a huge splash was made by AlphaFold in 2018.
Ah, yes, predicting protein folding. That was massive news.
Game-changing. Predicting how proteins fold into their 3D shapes with amazing accuracy. It has enormous implications for biochemistry, drug development, you name it.
It's quite a journey, from Dendrill's rules to AlphaFold's deep learning. Okay, let's get into the nuts and bolts a bit. What are the fundamental ML and deep learning ideas that chemists really need to grasp?
Well, the document starts with a core distinction: supervised versus unsupervised learning.
Right. Supervised is when you have labeled data.
Exactly. It's like learning with a teacher or, well, an answer key. You give the AI examples input features paired with the correct output or label. The AI learns the mapping.
Like predicting boiling point, where the boiling point is the label.
Precisely. Boiling point prediction is a regression task predicting a continuous value, or classifying if a compound is active or inactive against a target that's classification, predicting a category.
Okay, that makes sense. Learning from examples with answers. What about unsupervised?
Unsupervised learning is different. You give the AI data, but without any labels or answers. The AI's job is to find structure or patterns within the data itself.
So it's more exploratory.
Very much so. A classic example in chemistry is clustering grouping similar molecules together based on their properties, without knowing beforehand what the groups should be. Or dimensionality reduction, finding ways to simplify complex data.
Finding the hidden structure.
Okay.
Yeah. And the document mentions features, labels, models. Can you unpack those quickly?
Sure. Features are just the measurable inputs you feed the model. In chemistry, that could be molecular weight, polarity, maybe data from a spectrum, anything you can measure or calculate about a molecule.
Input data points.
Right. Labels, as we said, are the outputs you're trying to predict in supervised learning: bioactivity, toxicity, reaction yield, whatever it is.
The target prediction.
Yep. And the model is the algorithm itself, the mathematical engine that learns the relationship between the features and the labels. It learns from the training data, so it can make predictions on new, unseen data.
Got it. Features in, labels, sometimes for training, model does the learning. So what specific algorithms does the document highlight as fundamental?
It covers a few basics. For supervised learning, you've got simple linear models like linear regression for continuous outputs and logistic regression for classification probabilities. Easy to understand, good starting points.
Okay.
Then decision trees, they make predictions using a sort of flow chart of questions about the features. Quite interpretable.
You can see the logic.
Exactly. SVMs, support vector machines, are more complex, but often very powerful, especially with lots of features. And then those ensemble methods we mentioned, random forests, gradient boosting, which combine multiple models, often trees, for better performance.
Right, the combined approach. And unsupervised.
For unsupervised, key algorithms are for clustering, like k-means, which partitions data into a set number of groups, and DBSCAN, which finds clusters based on density and is good at finding outliers, too.
Grouping similar things, finding odd ones out.
Yeah.
Useful.
Definitely. Okay, digging a bit deeper into supervised learning regression methods.
Yeah, you mentioned linear, but also polynomial.
Right. Linear regression fits a straight line. Polynomial regression can fit curves, capturing more complex, nonlinear relationships between features and the output. And logistic regression, despite the name, is for classification. It predicts the probability of something belonging to a class, like active versus inactive.
Okay. And for classification specifically, besides logistic regression?
The document mentions SVMs again, often very effective when you have many features, common in chemistry for predicting bioactivity.
High-dimensional data.
Yes. KNN, K nearest neighbors, is interesting. It classifies a new molecule based on how its neighbors are classified in the feature space. Kind of like prediction by analogy.
If it looks like a duck, cracks like a duck.
Sort of. And naive Bayes is mentioned too. It's computationally fast and works surprisingly well, especially for things like text classification, maybe mining chemical literature.
Interesting. What about decision trees and those ensemble methods again? How are they used?
Decision trees are great for interpretability, but a single tree can sometimes overfit the training data, meaning it learns the noise, not just the signal.
Right, too specific.
Yeah, so ensemble methods help fix that. Bagging, like in random forests, builds many trees on different random subsets of the data and features, then averages their predictions. This makes the result much more stable and accurate.
So random forests are good for?
Things like predicting biological activity, screening potential materials, lots of applications.
Okay, and gradient boosting?
Gradient boosting machines, or GBMs, also build trees, but sequentially. Each new tree tries to correct the errors made by the previous ones. They can become extremely powerful, really good at capturing complex, nonlinear patterns, useful for prioritizing drug leads, for example.
So more sophisticated ways of combining trees. Got it. Now, shifting to unsupervised learning clustering and dimensionality reduction, why are these so key in chemistry?
Well, clustering, using methods like k-means or dbScan, is super useful for exploring large chemical data sets. You can automatically group compounds with similar properties. Maybe you find a new class of molecules, or maybe you spot an outlier compound that's really unusual and potentially interesting.
Discovering novelty or structure within the data.
Exactly, and dimensionality reduction. Chemistry data often has tons of features. Think of all the ways you can describe a molecule numerically.
Right. Gets complicated fast.
It does. Techniques like principal component analysis, PCA, help boil this down. They find the main axes of variation in the data, letting you represent it with fewer dimensions while keeping most of the important information.
Makes it easier to visualize and analyze.
Yes, and often improves the performance of subsequent machine learning models because you remove redundant or noisy features. Imagine trying to plot data in 500 dimensions. Impossible. PCA can help get it down to 2 or 3.
Makes sense. Are there other ways to reduce dimensions mentioned?
Yeah, the document mentions TSNE and UMAP, which are particularly good for visualization, creating those nice 2D maps where similar molecules cluster together visually. Also, autoencoders, which are a type of neural network used for dimensionality reduction.
Okay, and anomaly detection fits in here too.
Right. Finding the odd ones out. This can use statistical methods, distance-based approaches, or density-based ones like dbScan. It's useful for, say, quality control in manufacturing or identifying rare compounds and screening data.
Finding the unexpected. Cool. Okay, we can't really talk AI without diving into neural networks properly. What are the basics here?
Okay, neural networks. The basic unit is the artificial neuron, or node. It takes some inputs, multiplies each by a weight. Think of weights as measuring the connection strength, sums them up, and then passes that sum through an activation function. This function introduces nonlinearity, which is really important. It's what allows the network to learn complex patterns, not just linear relationships.
And examples of activation functions.
Common ones are sigmoid, which was used a lot historically, ReLU, rectified linear unit, which is very popular now because it's efficient, and softmax, often used in the output layer for classification tasks to get probabilities for each class.
Right, and these neurons are arranged in layers.
Usually, yes. The simplest is the feed-forward neural network. You have an input layer that receives the features, one or more hidden layers where the computation happens, and an output layer that gives the prediction. Information flows strictly forward from input to output.
Okay, simple structure. But how do they learn? How do those weights get adjusted?
That's the magic of backpropagation and gradient descent.
The terms we heard earlier.
Exactly. So the network makes a prediction, you compare it to the true answer, calculate the error. Backpropagation is the algorithm that efficiently calculates how much each weight in the network contributed to that error.
Working backwards through the network.
Precisely. Then, gradient descent is the optimization algorithm that uses that error information to nudge the weights, bit by bit, in the direction that reduces the error. You repeat this process many times with your training data, and the network gradually learns to make better predictions.
Learning by minimizing mistakes. What about overfitting, making sure it doesn't just memorize the training data?
Yeah, that's a big challenge. The model needs to generalize to new data. Regularization techniques help prevent overfitting. Dropout is a common one during training. You randomly switch off some neurons, forcing the network to learn more robust representations. L1 and L2 regularization add penalties to the weights themselves, discouraging overly complex models.
Okay, ways to keep the model honest, basically. Now, deep learning uses specific architectures. Which ones are especially relevant for chemistry?
The document highlights several. Convolutional neural networks, CNNs, are superstars for grid-like data, like images.
How does that apply to chemistry?
Think about analyzing microscope images of crystals or interpreting data from techniques that map chemical properties across a surface. You can even represent molecule interactions, like protein-ligand binding, as a sort of 2D or 3D map that a CNN can process.
Ah, okay, spatial data. What about sequences?
For sequential data, recurrent neural networks, RNNs, and their more advanced cousins like LSTMs, long short-term memory networks, are key.
Sequential data like smile strings.
Exactly. Smiles notation represents molecules as a string of characters. RNNs LSTMs can process these sequences to predict properties, or even generate new valid smile strings for novel molecules. They're also used for modeling reaction mechanisms, which unfold over time.
Generating new molecules, that sounds powerful. What about graph neural networks? Molecules are basically graphs, right?
Precisely. Molecules are naturally represented as graphs. Atoms are nodes, bonds are edges. Graph neural networks, GNNs, are specifically designed to learn directly from this graph structure.
What can they do?
They're incredibly powerful for predicting molecular properties directly from the graph. They learn fingerprints or representations that capture the connectivity and chemical environment. Also great for analyzing biological networks, reaction networks.
Seems like a very natural fit for chemistry.
It really is. Then you have generative models, like VAEs, variational autoencoders, and Jan's generative adversarial networks.
These are the ones that create new things.
Yes. VAEs learn a compressed representation, latent space, of molecules, and can then generate new ones by sampling from that space. Jan's use a clever two-network system. A generator creates fake molecules, and a discriminator tries to tell them apart from real ones. They compete and get better together.
So they can dream up completely new molecules.
Potentially, yes. Molecules that are hopefully realistic and have desired properties. It's a very active area of research for drug design and materials discovery. There are challenges, like ensuring the generated molecules are valid and synthesizable, but the potential is huge.
Wow. Any other key architectures?
The document also mentions attention mechanisms and transformers. These originally revolutionized natural language processing, but they're proving useful for chemistry too. They're good at handling long-range dependencies and sequences, so they can be applied to predicting properties from smiles or modeling reactions described textually.
Okay, an amazing toolkit.
Yeah.
But how do you know if your model's actually any good? Evaluation is key, right?
Absolutely crucial. You can't just build a model and assume it works. The document covers various evaluation metrics. For regression predicting numbers like boiling points, common metrics are mean squared error, MSE, root mean squared error, RMSE, and mean absolute error, MAE. They measure the average prediction error in different ways.
Okay. What about classification active-inactive?
For classification, simple accuracy, the percentage of correct predictions can be misleading, especially if your data set is imbalanced. You're doing way more inactive than active molecules.
Right. Predicting inactive all the time might look accurate, but miss all the important active ones.
Exactly. So you need metrics like precision, out of all predicted positives, how many were actually positive, and recall, out of all actual positives, how many did we find? The F1 score combines these two.
Balancing finding the right ones versus avoiding wrong ones.
Correct. ROC curves and AUC, area under the curve, and precision recall curves are also important for visualizing the trade-offs between true positives and false positives, especially with imbalanced data.
And how do you avoid just evaluating on the data you trained on?
That's where cross-validation comes in. You split your data into multiple folds, train the model on some folds, and test on the remaining one, then rotate. This gives a much more robust estimate of how the model will perform on unseen data and helps prevent overfitting.
Makes sense. Don't test on the answers you already saw.
Right. And then there's hyperparameter tuning, finding the best settings for the model itself, like the number of layers in a neural network or learning rate. Grid search or random search are common ways to explore different settings. It all ties into the bias variance trade-off. Trying to find a model that's complex enough to capture the patterns, low bias, but not so complex that it fits the noise, low variance.
A balancing act. Okay, this AI relies heavily on data. The document dedicates a whole section to data management. Why is that so critical?
Because data is the fuel, garbage in, garbage out, right? The quality, quantity, and accessibility of data are paramount for successful AI in chemistry.
So what kinds of chemical data are we talking about?
Broadly, the document groups it into experimental data, things directly measured in the lab, like synthesis details, results from spectroscopy, NMR, IR, mass spec, bioassay outcomes.
The raw lab results.
Yeah, and accurate documentation here is key. Then there's computational data results from simulations like quantum chemistry calculations, predictions from existing models, outputs from virtual screening experiments. This data often helps generate hypotheses.
Data generated by computers.
Right. And finally, data from public databases and repositories, huge collections like those from the NIH, the Royal Society of Chemistry, RSC, the Protein Data Bank for structures, the Materials Project for materials properties. Making data accessible is vital.
Okay, multiple sources. How is this data typically collected?
A major driver is high-throughput screening, HTS. This uses automation robots, microplates, to rapidly test thousands or even millions of compounds for activity. Think drug discovery or material screening.
Industrial scale experimentation.
Pretty much. Automation is key across the board. Now, automated spectrometers feeding data directly to computers, laboratory information management systems, LIMS tracking samples and results, even remote monitoring of experiments. It reduces errors, improves reproducibility.
Getting data automatically.
And another huge source is literature and patents. There's a vast amount of chemical knowledge locked away in text. Text and data mining, TDM techniques, including chemical named entity recognition, are used to extract structures, reactions, properties, and trends from papers and patents. Helps researchers keep up.
Mining the archives. Sounds challenging but necessary. So once you have the data, it's probably not perfect, is it? Pre-processing must be essential.
Oh, absolutely critical. Raw data is often messy. First step is data quality assessment, checking for errors, inconsistencies, outliers. You need validation rules, maybe quality metrics like signal-to-noise ratio.
Cleaning it up.
Yeah. Then handling missing data. Do you remove samples with missing values? Or try to impute them, estimate the missing value based on other data? Depends on the situation.
Tricky decisions.
Can be. Data normalization or scaling is also often necessary. Many algorithms are sensitive to the scale of features, so you often rescale them to, say, a range between 0 and 1, or give them a standard deviation of 1.
Putting everything on a level playing field.
Exactly. And finally, feature selection and engineering. Do you need all the features you collected? Maybe some are redundant or irrelevant. Feature selection techniques help pick the most informative ones. Feature engineering is about creating new, potentially more predictive features from the existing ones, maybe combining some or calculating a ratio.
Well, finding the inputs for the model. Got it. Now, with HTS and simulations, we're often talking big data in chemistry. How do researchers handle that scale?
You need specialized tools and infrastructure. For storage, things like large relational databases or increasingly data lakes that can handle diverse data types.
Storing massive amounts.
Right. For processing, distributed computing frameworks like Apache Hadoop and Apache Spark are key. They allow you to break down computations and run them across many machines in parallel.
Harnessing clusters of computers.
Yep. Parallel computing in general, often using cloud services like AWS, Google Cloud, or Azure, provides the necessary horsepower for training large models on big data sets.
Renting computing power.
Essentially.
Yeah.
And managing the whole flow data ingestion, cleaning, transformation, model training, deployment requires data pipeline management tools. Things like Apache NiFi or Airflow help orchestrate these complex workflows reliably.
Automating the whole data journey.
Pretty much. It ensures consistency and reproducibility.
The document also calls out specific databases. Which ones are particularly important for chemists using AI?
It lists several key ones. For chemical structures and general compound info, PubChem from NCBI and ChemSpider from RSC are huge, comprehensive resources. Essential for finding compounds, properties, links to literature.
The chemical encyclopedias?
Kind of. For bioactivity data, crucial for drug discovery and QSRR, ChemBel from MLEB and BindingBB are go-to databases. They contain curated data on molecules and their measured interactions with biological targets.
Data focused on drug action.
Yes. And for materials science, the Materials Project and the Open Quantum Materials Database, OQMD, provide vast amounts of computed properties for known and hypothetical materials, invaluable for searching for materials with specific functions.
Databases driving materials discovery. Okay. The last point in the data section is about sharing and reproducibility. Why the emphasis here?
It's fundamental for scientific progress, especially with data-driven science like AI, open science principles like publishing open access, sharing data openly, using preprint servers for rapid communication, accelerate discovery, and allow others to build on previous work.
Making science more transparent and collaborative.
Exactly. Standardizing data formats is also crucial. Using common formats like SDF or MOL or SMILES for structures or standardized exchange formats like ANML for analytical data makes it much easier to share and combine data from different sources. Metadata standards help describe the data properly.
So everyone speaks the same data language.
Ideally, yes. And reproducible workflows are key for trust. Using electronic lab notebooks, ELNs, to digitally record experiments, version control systems like Git for code and analysis scripts, and workflow management tools like Nime or Galaxy, these all help ensure that others can understand and replicate your findings.
Building confidence in the results.
Definitely. And finally, you can't ignore intellectual property. How do patents work with AI discoveries? How do you handle data licensing, privacy regulations like GDPR, data security? These are essential practical and ethical considerations when sharing or using data.
Lots to consider on the data front. Okay, let's move into how AI is actually being woven into different chemical fields, starting with computational chemistry, AI helping the helpers, so to speak.
Yeah, it's an interesting synergy. Computational chemistry itself uses methods rooted in physics, quantum mechanics, like Hartree-Fock, DFT, or faster semi-empirical methods, and molecular mechanics using force fields for huge systems like proteins. QMMM combines both.
Different tools for different scales and accuracy needs.
Right. And AI is now boosting these methods. For instance, calculating potential energy surfaces, how energy changes as atoms move, is vital but often very costly with QM. ML models, like neural networks or Gaussian processes, can learn to predict these surfaces much, much faster after being trained on some QM data.
So speeding up fundamental calculations.
Massively. AI is also used to improve the accuracy of methods like DFT by learning correction factors for the approximations used in standard DFT functionals. Some AI approaches are even trying to predict electron density directly, which is fundamental for reactivity.
Getting better accuracy or speed or both. What about molecular dynamics, simulating molecules moving over time?
Empty simulations track atomic motions. AI helps here too. Techniques like accelerated MD use machine learning to identify important slow motions or rare events, making simulations more efficient at capturing key processes.
Focusing the simulation on what matters.
Exactly. And a huge area is AI-derived force fields. Traditional force fields are fast but approximate. New methods like PhysNet or ANI use deep learning, trained on quantum data, to create force fields that are nearly as accurate as QM but orders of magnitude faster. This allows for very accurate simulations of larger systems for longer time.
Wow, bridging the accuracy-speed gap.
It's a major advance. AI also helps with enhanced sampling methods, techniques designed to explore the conformational space of molecules more effectively during simulations.
Okay. And predicting molecular properties is a core task. How's AI changing that game within computational chemistry?
It's becoming ubiquitous. For basic physicochemical properties, log-P for lipophilicity, pKa for acidity, solubility, ML models, especially GNNs, are often outperforming older methods.
Predicting fundamental chemical character.
Yep. Same for spectroscopic properties. Predicting NMR chemical shifts, interpreting complex IR or Raman spectra, predicting UVV's absorption. ML models trained on large spectral databases can do this rapidly and often very accurately. Pattern recognition is key here.
Learning the structure-spectrum relationship.
Precisely. And for thermodynamics and kinetics, predicting reaction energies, activation barriers, rate constant, AI models are being developed to provide quick estimates, complementing or sometimes replacing more expensive QM calculations, especially for initial screening.
Getting quick answers for energy and speed of reactions.
Right. The document gives some case studies too, like AI-assisted catalyst design.
How does that work?
Using ML for high-throughput virtual screening of potential catalyst materials, predicting their activity, or even inverse design telling the AI you want a catalyst for a specific reaction with certain efficiency. And it suggests structures, big implications for energy applications, green chemistry.
Designing catalysts on demand. Cool. What about reaction pathways?
Understanding how a reaction proceeds step by step is complex. AI tools are being developed to automatically generate plausible reaction pathways. Then ML models can estimate the kinetics, the energy barriers for each step, helping chemists understand the mechanism or design better synthetic routes.
Unraveling the reaction mechanism with AI help.
Exactly. And computational design of functional materials is another big one, using AI to predict properties like conductivity, strength, optical response for materials like solar cells, superconductors, polymers. Again, using ML for screening and inverse design to accelerate discovery.
Finding the next generation of materials. It's clear AI is deeply embedded in comp chem now. Let's shift to pharmaceuticals drug discovery. This seems like a prime area for AI impact.
Oh, absolutely. It's transforming nearly every stage, starting right at the beginning. Target identification and validation.
Finding what to aim the drug at.
Exactly. AI analyzes huge omics data sets, genomics, proteomics, etc., looks at biological networks, patient data, to identify proteins or pathways involved in a disease that could be good drug targets, finding biomarkers too.
Sifting through biological data for clues.
Right. Then comes finding the actual drug molecules, hit identification and lead optimization. Virtual screening is a mainstay here.
Screening compound libraries computationally.
Yes. Ligand-based methods use known active molecules to find similar ones. Structure-based methods use the 3D structure of the target protein, often involving docking simulations. AI is enhancing both better classification models for ligand-based screening, AI-powered scoring functions to improve docking accuracy. Hybrid methods combine approaches. Active learning uses AI to intelligently select which compounds to screen next.
Making virtual screening smarter and faster.
Definitely. And then there's de novo design AI actually creating new drug candidates from scratch.
The molecule generation we talked about.
Exactly. Using those generative models, RNNs, VAEs, YANs, often operating on smiles or similar representations, to invent novel molecular structures. Reinforcement learning is often added to steer the generation towards molecules with better predicted properties. Potency, low toxicity, ease of synthesis, juggling multiple objectives.
AI is a molecular architect.
A pretty good analogy, yeah. Then, once you have candidates, you need to predict how they'll behave in the body, AD-MET properties.
Absorption, distribution, metabolism, excretion, toxicity, the crucial factors.
Right. Will the drug get absorbed? Where will it go? How will it be broken down? How will it leave the body? Will it be toxic? Predicting these early is vital to avoid costly failures later in development. AI models, both classification and regression, are being built to predict all these properties based on molecular structure.
Predicting drug fate and safety before animal or human tests.
That's the goal. Reducing reliance on experimental testing, speeding things up, picking winners earlier.
Are there real-world successes yet? AI-designed drugs in the clinic.
Yes. Things are moving fast. The document mentions examples like DSP-1181 from Exscientia, which entered clinical trials. In Silico Medicine is another company reporting AI-discovered drugs moving into development. We're seeing more pharma companies partnering with AI specialists. AI is also impacting personalized medicine, tailoring drugs based on individual genetic profiles.
So it's starting to deliver tangible results.
Yeah.
What about the regulators like the FDA or EMA? How do they view AI in drug development?
They're actively engaged. Agencies are issuing guidelines on using AML. Key concerns are transparency. Can we understand why the AI made a prediction? Validation, how rigorously was the model tested? And data integrity. They need robust standards, good machine learning practices, GMLP, clear documentation.
Ensuring the AI tools are reliable and the results trustworthy.
Exactly. Ethical considerations are also huge potential biases in the data, leading to biased predictions, data privacy, patient consent, all critical issues.
Okay, a complex but rapidly evolving landscape in pharma. Let's switch to material science and nanotechnology. Similar impact there.
Very much so. The whole idea of the Materials Genome Initiative, for example, is heavily reliant on data and computation, including AI, to accelerate materials discovery.
Applying the genome idea to materials.
Right, using ML to predict material properties, electronic, mechanical, thermal, optical from composition or structure, screening databases, identifying correlations. Inverse design is big here too. Specify the properties you want, and AI suggests materials.
Designing materials for function.
Exactly. In nanotechnology specifically, AI helps characterize nanomaterials, analyzing complex electron microscopy images, interpreting spectroscopic data from tiny samples. It's also used to predict potential nanotoxicology risks and to optimize the synthesis processes using AI with design of experiments, DOE, or process process modeling for better control and scale up.
Managing the unique challenges of the nanoscale.
Yes. And high-throughput screening, like in pharma, is used here too. Combinatorial chemistry creates libraries of materials, automated platforms test them, and AI analyzes the resulting flood of data to find the needles in the haystack.
Automating the materials discovery pipeline.
Pretty much. The document gives case studies, again, using AI in the search for better superconductors, designing new polymers with specific properties, creating photonic materials that manipulate light in novel ways.
Real examples of AI driving innovation.
And a lot of focus is on applications and sustainability, finding better materials for solar cells, improving battery technology, discovering new catalysts for cleaner energy production or environmental remediation. AI is a key tool in that effort.
Using AI for greener technologies. Great. Okay, let's talk chemoinformatics. It feels like this field is AI applied to chemical information.
There's huge overlap, definitely. Chemoinformatics is all about managing and analyzing chemical information using computational tools, representing molecules, smiles, fingerprints, 3D coordinates, managing databases, searching chemical space. AML methods are central tools for chemoinformaticians.
So what are the key ways molecules are represented for AI?
Lots of ways. Simple text strings like smiles or inchi. Molecular fingerprints are very common. These are bit strings or vectors that encode structural features. There are different types. Topological circular fingerprints like ECFP. They turn a structure into a numerical format ML can understand. 3D descriptors capture shape. Pharmacophores represent key interaction points for drug binding.
Different ways to turn a molecule into numbers.
Essentially, yes. And managing chemical databases and compound libraries is core chemoinformatics. Using ML for mapping chemical space, clustering compounds to understand diversity or select subsets for screening, reducing redundancy, all standard tasks.
Organizing the world of known chemicals.
Right. And QSIR, which we touched on, is a classic chemoinformatics application that heavily uses machine learning now.
Building those structure activity models. What's new there?
Well, moving beyond traditional linear models like multiple linear regression, PLS, to more powerful nonlinear ML methods, neural networks, SVMs, random forests, deep learning gives much more predictive power, especially for complex biological endpoints. But validation is even more critical with these complex models. Defining the applicability domain where the model can be trusted is essential, especially if you want to use these models for regulatory purposes, like under REACH regulations, as potential alternatives to animal testing.
Ensuring that predictions are reliable and used appropriately.
Exactly. The OECD principles for QSR validation are key here.
What about predicting reactions themselves?
Another big area, predicting the products of a reaction given the reactants and conditions. Older systems were rule-based, limited by encoded knowledge. Newer methods use sequence-to-sequence models, like in translation, or GNNs to learn reaction patterns from large data sets like the USPTO patent database.
Learning chemistry rules from data.
Yes. Also predicting reaction yield or selectivity. And AI-assisted reaction optimization using Bayesian optimization or reinforcement learning to suggest the best conditions, temperature, solvent, catalyst, to maximize yield or purity.
Fine-tuning reactions automatically.
That's the idea.
Yeah.
And retrosynthesis planning, which we mentioned with LH-OSA, AI is tackling that too. Rule-based systems still exist, but neural approaches, SIP2-seq graph-based, are learning complex synthesis strategies directly from reaction data, sometimes suggesting surprising or more efficient routes. Integrating these with knowledge bases makes them even smarter.
AI is a synthetic chemistry assistant.
A powerful one, potentially. Okay, last integration area. Spectroscopy and analytical chemistry. How's AI helping interpret all that spectral data?
Right. Spectra are data-rich but complex.
Exactly. AI is used for basic processing, noise reduction, baseline correction, peak detection, and assignment automating tedious tasks. It's used for multivariate analysis, pulling out subtle patterns from complex spectra like NMR or mass spec. Signal enhancement, too.
Cleaning up and digging deeper into spectra.
Yes. Specifically for NMR, AI helps automate structure elucidation workflows, predicts chemical shifts very accurately, often combining ML with QM, and even analyzes dynamic NMR to understand molecular motion.
Decoding NMR signals faster.
For mass spectrometry, ML helps deconvolute complex spectra, identify metabolites and metabolomics studies, and interpret the massive data sets from proteomics experiments.
Making sense of mass spec fragmentation patterns.
Right. In IR and Raman spectroscopy, AI powers spectral library searching and pattern recognition for identifying substances. It's also key in advanced techniques like SERS, surface-enhanced Raman, for interpreting enhanced signals or analyzing hyperspectral imaging data, where you get a spectrum at every pixel.
Pulling chemical information out of images.
Exactly. AI is also huge in microscopy image analysis, generally noise reduction, feature segmentation, 3D reconstruction from 2D slices. In integrating data from multiple techniques, correlative spectroscopy and imaging AI helps fuse that information.
Bringing different analytical views together.
Yes. And even in electrochemistry and sensors, AI helps process noisy signals, calibrate sensors, and correct for drift over time.
Improving sensor reliability. Phew, that's a massive range of applications. But it can't all be smooth sailing. What are the big challenges and limitations the document points out?
Definitely challenges remain. Data is a big one. Often there isn't enough high-quality, well-labeled data for specific chemical problems, data scarcity. Data can also be very heterogeneous, coming from different labs, different techniques, making it hard to combine. We need more data sharing, better standardization.
The data bottleneck.
It's often the biggest one. Computational cost is another factor. Training large deep learning models requires significant computing power and resources, which isn't available to everyone.
The compute barrier.
Right. Integrating AI into actual experimental workflows is also non-trivial. There's often a gap between an AI prediction and easily translating that into a lab experiment. Closing that loop, having experiments feed back into the AI is key.
Bridging the gap between digital and physical.
Exactly. Bias is a huge concern, too. Bias in the data collection, bias from historical inequalities reflected in the data, even algorithmic bias. This can lead to models that perform unfairly or unreliably for certain types of molecules or problems. Careful data curation and bias mitigation strategies are crucial.
Ensuring fairness and reliability.
And finally, interpretability. Many powerful AI models, especially deep learning ones, are black boxes. It's hard to understand why they make a particular prediction. This lack of transparency can be a barrier to trust and adoption, especially in high-stakes areas like medicine or regulation. Explainable AI, XAI methods are being developed to address this.
Opening the black box. Okay, those are significant hurdles.
What?
What about ethical considerations more broadly?
Very important. The document raises concerns about the potential misuse of AI in chemistry. For instance, could AI be used to design or synthesize dangerous substances like novel illicit drugs or chemical weapons? This raises questions about access control and responsible development.
The dual-use problem.
Exactly. Dual-use technologies that can be used for good or harm. Biosecurity is another related concern, especially with AI applied to genomics and synthetic biology. There's a need for strong ethical guidelines, international cooperation, and researchers taking responsibility. Frameworks emphasizing fairness, accountability, transparency, and privacy are essential.
Thinking through the societal impact.
Absolutely. And related to the black box problem is the need for model transparency, especially for regulatory submissions. How do you convince regulators a drug designed or tested using AI is safe and effective if you can't fully explain the AI's reasoning? Developing standards for validation and building user trust are key.
Making AI accountable.
Yes, and legal implications too. Who owns the intellectual property, the patent for a discovery made by an AI? The programmer. The user, the AI itself. Current patent law wasn't designed for this. Data protection laws like GDPR also apply when using sensitive chemical or biological data. Standards for AI validation and maybe even certification will likely be needed.
New legal and regulatory frontiers. So looking ahead, despite the challenges, what does the future hold? What's exciting?
The document paints a picture of increasing automation. Think self-driving labs or autonomous discovery systems, where AI designs experiments, robots execute them, AI analyzes the results, and the loop repeats with minimal human intervention. Closed-loop systems.
A lab of the future.
Potentially. Integration with other technologies, like quantum computing, could unlock currently intractable chemical problems. Personalized and precision chemistry drugs and materials tailored to individuals or specific needs seems more achievable with AI.
Custom chemistry.
Right. And a big push towards sustainable and green chemistry. AI can help design better catalysts, find greener reaction pathways, optimize processes to reduce waste and energy consumption.
AI for a greener planet.
Hopefully. And the role of the chemist evolves, too. It becomes less about routine tasks and more about collaborating with AI, asking the right questions, interpreting complex results. Leveraging AI as a powerful tool, it requires new skill sets.
Chemists becoming AI partners. Okay, for someone listening, maybe a chemist or student who's thinking, okay, I want to try this, what are the first steps?
Define your problem clearly. What chemical question are you trying to answer with AI? Collect and prepare your data carefully. This is often the hardest part. Then start exploring the tools. Python is the dominant language, with libraries like NumPy, Pandas for data handling, scikit-learn for general ML, TensorFlow and PyTorch for deep learning. Chemistry-specific toolkits like ArtyKit or OpenBabel are essential for handling molecular data. Visualization tools like Matplotlib or Seaborn help understand results.
Learning the tools of the trade.
Yes, building interdisciplinary teams is often key chemists collaborating with data scientists or computational experts. Allocate resources, compute time, software, maybe training. And start small, manage risks, maybe tackle a simpler project first.
Don't try to boil the ocean on day one.
Exactly. The document even suggests hands-on examples like predicting boiling points, calculating molecular similarity, maybe building a simple neural network, designing a workflow for a specific task, thinking about the real-world application and potential return on investment.
Practical starting points. Well, this has been a truly comprehensive tour of AI in chemistry, drawing heavily on that Gaber document. It's undeniable that AI is reshaping the field.
It really is. And for you listening, the key takeaway is how AI is amplifying our capabilities across the board: discovery, design, analysis, automation. It's a force multiplier.
But also remembering the limitations, data quality, the ethical dimensions, the need for collaboration and careful validation. It's not magic.
Not magic, no. A powerful tool that needs to be wielded wisely.
And thinking about those specific applications, the idea of AI designing molecules that actually reach clinical trials or helping find materials for sustainable energy, that's pretty mind-blowing stuff. It shows the real-world potential.
Absolutely. It brings it home. And maybe that leaves you with a final thought to chew on. What fundamental chemical questions, maybe ones we haven't even dared to ask properly before, might become solvable as these AI tools get even more sophisticated? What new kinds of chemical understanding could emerge from this powerful combination of human and artificial intelligence?
A fascinating question indeed. What new chemistry will AI help us uncover? Lots to think about.